Welcome dear students for HSC online lecture initiative taken by Shet Vidya Mandir English High School and Junior College of Science and Commerce. I am Miss Ragini Singh, Assistant Lecturer of Biology, and today we are going to start with a very new chapter, that is Enhancement in Food Production, and today's topic is plant breeding. Now before I start with the lecture, I request you all to please be seated with the notebook, your textbook and the stationery, that is your pen and pencil, so that you can jot down the important points. Let's revise something that will be related to this chapter. I will be giving you the photos of the hybrid plants. You need to identify which type of hybrid plants are they. Look at this first picture and look at this second picture. Now in the first picture you can observe that on the shoot part there is the formation of the tomato, whereas at the root part there is the formation of the potato. It means this plant is a combination of potato as well as tomato. This is one of the very unique method, and in addition to that it is also going to save a lot of space, and this combination, that is the hybrid combination of the potato and tomato, is called as pomato. Whereas if you observe the second diagram, here you can see the structure of the cabbage as well as the leaf structure and color of the radish. So this is the combination of radish and cabbage, and it is called as rabbage. So these are the few examples of the hybrid plant. Hybrid word itself is very self-explanatory, which means it's a combination of the two different things. What is plant breeding? Now when you talk about the plant breeding is the improvement or purposeful manipulation in the heredity of crops. Of course, if you want to bring out the new variety or the new changes in the particular crop, you need to work at the heredity level, that is at a genetic level, and the production of new superior varieties of existing crop plants. This constitute plant breeding. Now the definition you need to learn and understand point to point from the textbook point of view. So I hope the definition of plant breeding is clear to you. Identify the scientist. The scientist is Dr. Norman E. Borlaug, and he is Dr. M.S. Swaminathan. Now both of the scientists are the father of the Green Revolution, and Dr. M.S. Swaminathan, to be very specific, he is the father of Indian Green Revolution. Both of them have contributed extremely well towards the field of agronomics. We need to understand the hybridization and its technique. Now this hybridization and techniques involve different steps. The main steps of this plant breeding program are as follows. The very first step is collection of variability. It means you are going to collect the plants of the different varieties such as disease resistance, good quality of the seed produced, good quality of the fruit produced, pesticide resistance, the plant which will be able to tolerate various environmental conditions. That is nothing but it is the collection of variability. The entire collection having all diverse alleles. The entire collection having all the diverse allele, that is the variations for all the genes in a given crop, is called germplasm collection. Very important word, that is germplasm collection. Now this can be done via two ways. One is in situ conservation, another is ex situ conservation. In situ conservation involves with the help of the forest and natural reserves. Suppose there is one particular plant which is going to be extinct, or it has already been extinct. Now you want it again because of its medicinal importance. What you can do, you can go in the forest, collect its wild form and then convert it into the domestic form via converting with respect to its heredity. Whereas when you talk about the ex situ conservation, it is done through this botanical gardens. The botanical gardens consist of many indigenous plant and it is very useful for us. And finally is the seed bank. The same way that we have the sperm bank, pollen bank, and we have the seed bank. Here if you want any particular crop of the particular quality, you can go to the seed banks and you can ask for that. So this is all about the collection of variability, very first step in the plant hybridization technique. Let's see what is the second step. The second step is evaluation and selection of parents, which involves which quality you want in your coming F1 generations. So the selected parent must be very healthy, they should be vigorous, and they should show desirable but complementary features. The selected parents are then sold for 3 to 4 generation to make them pure or homozygous. Homo means same. If you want your future generation, that is your offspring, to be of the desirable character of the parents, you need to self them for 3 to 4 generations. Now look at this plant. This is plant A and this is plant B. Now you yourself tell that which plant you will be going for as a parent plant. Of course you will be going for this plant A and not for the plant B. The very but obvious reason, because the plant A is very healthy and it is going to give rise to a very good offsprings. It is made sure that the pure lines are selected, multiplied and used in the hybridization, which is the third step. So the third step includes hybridization, which means crossing between two plants having different genetic makeup. Now in this hybridization, the pollen grains from anther of male parent are collected and then they are artificially dusted over stigma of the emasculated flower of the female parent. Now what is this emasculation? Emasculation is removal of the anther before anthesis, and this is possible only in case of bisexual flower. See, this is a bisexual flower which consists of the gynoecium part as well as the androecium part. Now I want the suitable pollen grain, that is of my desirable quality, to be placed on this stigma. So for that I need to remove this anther, and once the anther is removed, it means once the emasculation is done, I can transfer the pollen of desirable quality over this stigma, and this process is called as the cross pollination. And once the pollination is done, I would be getting the F1 hybrid. Why the word hybrid? Because it is of the two different genetic makeup mixed together to give you a F1 hybrid. So this is about hybridization, which involves cross pollination. The F1 hybrid progeny is selected and evaluated for desired combination of characters. The fourth step is selection and testing of superior recombinants. Now I have already received the F1 generation. From that F1 generation I am going to select and test the superior recombinants. Now look over here. The F1 hybrid, it shows the superiority over both the parents. It means if suppose the plant A is very disease resistant and the plant B is producing many desirable fruits of good quality, it means the F1 plant, that is the F1 progeny, which I would be getting, will be showing both the quality that is of the parents, and this means they are having the high hybrid vigor. Such hybrids are then sold for few generations to make them homozygous for the desirable characters till there is a state of uniformity, so that the characters will not get segregate or they will not get separate further. Look at this selection and testing of superior recombinants. Over here the selection is done as well as the testing is done of the superior recombinant to make sure that the recombinant is having the desirable characters. Self-pollination is needed to achieve the homozygosity. So this was the fourth step, that is selection and testing of the superior recombinants. Your last step is the testing, release and commercialization of the new cultivars. What you are supposed to do in this step is that the newly selected lines are evaluated for the productivity and other features like disease resistance, pest resistance, quality, etc. The selected lines are grown for 3 generations at least in natural field in different agroclimatic zone. This agroclimatic zone is very important because all the plants are not subjected to only one climatic condition. They require different types of climatic conditions. Finally, the variety is released and the new variety for use by the farmers. Here the testing is done first in the research field, which is grown for the 3 seasons, that is the 3 consecutive season it is grown. It is checked in for the various agroclimatic zone, and finally the new cultivation is done. On the left hand side, if you observe carefully, this is a plant tissue culture laboratory. So this was about the testing, release and commercialization of new cultivars. Let us see this with respect to a flow chart. Here you can see the very first step, collection of variability either from the germplasm or gene bank. Second step is you have to evaluate and do the selection of the parents. Third is selection of the parent plant with different qualities. You have to self the selected plants for at least 3 to 4 generations to bring the homozygosity. Then you have to identify the parents as a male parent and female parent. Collection of pollen from the flowers of the male parent. The next step is emasculation, and once this emasculation is done, you can go for the cross pollination, and once this cross pollination need to be achieved, these are the 2 important steps to be done, that is bagging and tagging. Now once the emasculation is done, here you can see the anther has been removed with the help of forceps. It means now it is not going to be pollinated by the self anther. It means autogamy is prevented, but there is chances keep cross pollination of any of the other pollen grain take place. Now if I want to avoid that, I need to do the process of bagging, in which already the emasculated flower has to be bagged. Once the bagging takes place, of course the exchange of air is taking place, but the pollen grain, that is the undesirable pollen grain, will not be sitting on the stigma, and if that is achieved, the cross pollination with respect to undesirable pollen grain can be prevented. Once the bagging is done, the next step is tagging. You need to tag this with respect to a paper, write down when the tagging was done and all the necessary detailed information. Once emasculation, bagging and tagging is done, there is the development of fruit and seed, that is F1 generation will be developed. Once the F1 generation is developed, you need to test the F1, and finally the F1 generation will be released for the field trials for the yield of productivity. So these are the very important steps for the plant tissue culture technique, especially for the hybridization technique. Now let us understand the Indian hybrid crops. If you look at the population of the India, it is growing at par. The crops that is being produced is not sufficient to fulfill this overgrowing population. That's why scientists have come with this hybrid crop technique. The very first hybrid crop technique that we are going to study is with respect to wheat and rice. Dr. Norman E. Borlaug in the 1960s, he developed 2 semi-dwarf varieties of wheat. The first one is Sonalika. This is a Sonalika semi-dwarf variety, and the second is Kalyan Sona. Now this both the variety are good with respect to the disease resistance or pest resistance. So the 2 varieties that was discovered or it was made is Sonalika and Kalyan Sona. Semi-dwarf rice varieties were taken from IR8, that is International Rice Research Institute, and Taichung native first from Taiwan, and it was introduced in India. And the semi-dwarf variety rice names are, this is Jaya, very simple name to remember. This is Ratna, and the third one is Padma. So remember semi-dwarf rice varieties Jaya, Ratna and Padma. sugarcane. Now when you talk about the sugarcane, we know that sugar is basically prepared from the sugarcane. This means sugarcane should produce large quantity of the juice so that the sugar can be made. Now Saccharum barberry is a native of North India and Saccharum officinarum, it belongs to South India. Both the Saccharum barberry and officinarum are in the group of the sugarcane. Now what is the scenario over here? The Saccharum officinarum, which belongs to the South India, they have a thicker stem and high sugar content, but it does not grow well in the North India. This is Saccharum barberry, native to North India, and this is Saccharum officinarum, that is in South India. Now when this Saccharum officinarum was subjected to grow in the North India, it was not able to grow because of the difference in the climatic zone. So what did the Sugar Breeding Institute, that is SBI, did? They prepared the hybrid varieties, and it was prepared in the Coimbatore, that is Tamil Nadu, by Sugar Breeding Institute, and they named it as Coimbatore 419, 421 and 453. Now to remember this number is also very easy. See, there is one number called as 420. Now 420 number is not there. One number before it, that is 419, and one number after it, that is 421. So this is the three varieties of this sugar cane which was produced, and thus this can be grown in the North India, even in the other parts of our country. Thus they would be yielding high sugar contents and the sugar demand could be easily met. The next is millets. Now what are the millets? Millets are nothing but they are the maize, jawar, bajra. Now the hybrid variety was grown with respect to the name, that is Ganga 3 for hybrid maize, jawar, that is CO12, and bajra, that is Nifad. Nifad is a place near the Nasik and itself means mountainous district, and the name has been given because it was grown over there. Now when you talk about this hybrid maize, this is Ganga 3, this is jawar, and this is bajra, that is Nifad. And these varieties are high yielding as well as resistant to water stress. It means if due to some reason there is not sufficient water available or the rain was not sufficient in the particular year, still this maize, jawar and bajra can be grown because of the resistance to the water stress, and it could meet the demand of the growing population. It is even the boon to the Indian farmers. So this is all about the millets. I hope this module is very clear to you, because now it's a time for you all to test. Test your brain. Your very first question is define emasculation. Now you can take your own time, pause the video, write the definition in your notebook and then check it out. Emasculation is nothing but it is the removal of anther before anthesis. Your second question is why Saccharum officinarum is superior over Saccharum barbaris? And answer is because it contains good sugar content as it has a thick stem. Your third and the last question is give examples of in situ and ex situ conservation. And the answer to this is in situ involves forest reserves as well as natural reserves, and ex situ involves seed banks and botanical gardens. Now one of the very interesting part of this, and that is your assignment. Your very first assignment question is describe briefly various steps of plant breeding methods, and second is enlist genetic hybrid varieties of rice and wheat. This both the assignment question you are going to write down in a proper note in your notebook, so that it will be very easy to assess and you will also understand what are the important points that to be written down in the exam. I hope this module was very helpful to you. For this assignment question you can refer your 12th standard textbook for the easy access. Thank you for attending this module. We will be coming with the module part 2 of this chapter. Thank you.